In this episode, we will talk about how to mimic human metabolism in mice.
For example, neurons, glia.
Let's talk pharmacokinetics for antibody development.
Following the transplantation of hematopoietic cells.
Let's talk how to test if your drug induces a cytokine storm.
Welcome to Jack's Tech Talk with the technical information scientists of the Jackson Laboratory.
Oh, hi, everybody. We are at SfN 2022, and I'm very happy to meet up with Mike Sasner from the Jackson Laboratory and Model AD Consortium, and he is going to tell us a little bit about his research. So I wanted to know, you've developed so many mouse models in the past 5 years. Which of those mouse models do you think is promising? And can you talk a little bit about that?
Well, first of all, it's always the next one, but the one we're focused on right now, we're really excited about, is what we're calling LOAD2, which is a humanized APOE4 allele, homozygous, humanized A beta allele, and a TREM2R47H risk allele, TREM. And on its own, it really doesn't have much of a phenotype, but when we put it on a high-fat diet, we realize mice normally get very healthy vegan diets, unlike you and me, for me anyway. So we put this on a high-fat, high-sugar diet, and we're actually seeing some really interesting phenotypes that look like early stages of Alzheimer's disease. Meaning we see changes in biomarkers, we see some neuroinflammation, we see some proteomics changes that mimic what we see in the human patients, and we're starting to see even some neuronal cell loss, specifically in females, which is really interesting, specifically in the cortex. So we're aging those mice. This is at 12 to 18 months of age. We're going later now, but we're really excited that we think this would be the first model that's really a good model for some parts, not all aspects, but certain parts of late onset Alzheimer's disease. And again, this is the current model where we're now moving forward and adding a humanized tau in that. So the Model AD program just got renewed for another 5 years, and a big part of that is adding humanized tau in there, so that we'll hopefully get not just amyloid type phenotypes, but also tau phenotypes as well.
Now, when you say humanized A beta, and when you say humanized tau, is wild type, right? Wild type version of it.
For these projects, yes, we're talking about non-mutant A beta, or specifically the A beta part of the APP gene, as well as tau. Although we do have mutations in both those genes, in the humanized versions of those genes. And in the next round of the renewal of Model AD, we're actually adding in some TREM2 variants that drive frontotemporal dementia or are associated with Alzheimer's disease. Because we really think it's important to, like I said, not just the amyloid pathology, but also the tau pathology.
Right.
And we think we need humanized tau. We know we need humanized tau, but we may also need some of the risk variants in the tau gene. So.
And at what age do you start that high fat plus sugar,
Both,
correct?
yes, yeah.
At what age do you start them on this, and for how long?
Generally, we start these at 2 months of age.
Okay.
And like I said, we go to 12 or 18 months.
Awesome.
Yeah.
Awesome. That's very exciting, because it seems a lot more translatable than the fast familial versions of it.
Yeah. And again, it's not Alzheimer's disease. It's some aspects of Alzheimer's disease, including some A beta 42 insoluble in serum, which is really exciting. I should point out we don't get amyloid plaques. We don't get pathology for amyloid. So it's not by any means a full amyloid model, but we think it might be what a person would look like at 40 years of age instead of 80 years of age, for example.
Right.
Which is really where we need to do the study. Right, we don't want to be studying late onset, we want to study early onset.
And have you used that model, the LOAD2, and tried different therapeutics with it with your pipeline, or not?
We are in the process of testing aducanumab, which is recently, it's been in trials for a long time. It's recently controversially, I should say, but it's been approved. So yeah, we have that drug in that mouse right now.
Nice.
So we're excited.
And at what age did you start it on LOAD2?
The diet, again, is 2 months, but the drug was at 6 months.
6 months?
Yeah, so going 6 to 12 months.
Okay.
Yeah.
So before any phenotype?
Yeah.
Okay.
Yeah.
Awesome.
Yeah.
Very curious to see what happens with that.
We're excited.
We'll follow up with you later.
That's a big part of the Model AD renewal is testing drugs that are either going to be in the clinic or in clinical trials right now in these mouse models.
And the prediction is that, because I'm not sure what it does in humans as far as does it decrease inflammation? What is, because I read that it is really more inflammatory in some cases, so I'm not sure what are the
Neuroinflammation is a big part of Alzheimer's disease, but the drugs seem to they don't improve things, but they stabilize things. So a patient doesn't get better, but they get worse less rapidly.
Okay.
That makes sense. Or they basically they plateau. Exactly.
Okay.
They plateau in a lot of different measures, which again it's very exciting that we can do that, but it's by no means making patients better. It's just
stopping the progression.
Stopping the progression. Yeah.
Awesome.
And the problem with that is, of course, we don't really know who's going to get Alzheimer's disease. Well, we're working on imaging and as a field are working on imaging and biomarker techniques to do that. But we're not there yet. But that's the goal, certainly, is to be able to work in the early stages and then just
Awesome.
Yeah.
And I know that your group has tons of posters. Do you want to tell us a little bit about the most exciting ones or the ones that have drawn more attention with viewers?
Yeah. So many pictures. I guess the one I'm most excited about is we have a primary screen. So we took a base model like LOD2 and we put 10 different risk variants. So these are not like APP or presenilin mutations to cause Alzheimer's disease. They're risk variants that increase your odds of getting Alzheimer's disease, as shown in human genetics and genomics. So again, we don't expect these to have full-blown Alzheimer's disease. We think they will push the mouse in Alzheimer's-like phenotypes. And we're testing these based on largely proteomics and transcriptomics.
Okay.
So it's very quantitative. And some of them look good, some of them don't look good, but then the ones that look good, we really focus down and then we do those to later ages and learn to put those on diet and other things. So, yeah, and actually we've picked 3 different variants, one in ABCA7, one in MTHFR, and one in PLCG2, and we've actually gone ahead and made those combinations, the complex 3 or 4 allele combinations. We're aging those out right now, to 18 to 24 months.
Nice.
So we're doing full-blown neuropathology, biomarkers, proteomics, metabolomics, and in vivo imaging.
Very cool.
So
Very
we
cool.
hope that people can then use those measures and say, okay, if I want a mouse model for my study, I know exactly not just which mouse model to use, but which phenotypes to look at and what age windows to look at, what therapeutic window you want to test the drug in, and then when you want to do the assays.
Right, and this is grant funding from NIH, correct?
This is all from the National Institute of Aging, yeah.
Okay.
Yeah. So we were lucky to just get renewed for that 5 years, and we're really excited to get that going. These are long experiments. If you have to create a combination of 3 or 4 alleles. That's months of breeding, generations breeding, and then, like I said, we're going 18 to 24 months of age. So these are long experiments.
Very long, very long. And the nice thing is that you are making all of these models available to the general researchers, right? To everybody once they are done.
They're all, no, before they're done. I mean, as soon as we make
the different pieces, right?
Well, but also the combinations. Everything we have, we will make available through Jack's Mouse Repository. Again, before we're done testing them, so people can take them and run with them, even before we have a lot of data, which is a blessing and a curse. People ask what the phenotypes are, and we say we don't know yet, but go ahead and run with them and do what you want.
Right.
And so the mice are all available from Jack's. It's important to point out these are available to biotech and pharma without any restrictions. In the past, a lot of them
as far as licensing, things like that.
In the past, the field has been littered with IP problems and royalties and things like that, and part of our mandate from the NIH was to make models that are not restricted anyway, so that biotech and pharma can use them because they're the ones testing the drugs, creating the drugs. Yeah.
So
and also I'll point out that all the data is available through the 80 Knowledge Portal, and so we have really 2 interfaces. 80 Knowledge Portal is a way to download all the data we have, then you can reanalyze it or do whatever you want. We also have the Data Explorer, which is more of a user interface. Type in your gene of interest, type in your model of interest, tissue of interest, whatever, and you can look at just your small set of data that you're focused on. So you don't have to be a bioinformaticist, whereas downloading from the 80 Knowledge Portal, you really have to know how to deal with big data sets.
Right.
I mean, these are transcriptomics, proteomics, they're huge data sets.
Yeah.
So
that's a fantastic resource.
Yeah, and then all our protocols are available. So we really want to make this like a cookbook, so someone can come in, order the mouse and Jacks. You know what age to look to do treatments. You know what assays to use for readouts, and really make it as as streamlined as possible.
And I've seen the protocols. They're very, very detailed protocols.
Yeah, it's a complex process, but
Yeah.
we want to share what we have as best we can.
That's amazing.
And along the same lines, I want to point out we have a weekly course every spring, basically teaching people, and this is mostly postdocs, grad students, some young faculty, how to do preclinical testing in mouse models.
Yeah, I did that
Disease.
course and I highly recommend it. It's amazing. It's amazing. Not only the course, the workshops, but all the discussions that happen, before and after classes.
Yeah.
It's amazing. I learned a ton.
We
during
have the whole
workshop.
lot of AD faculty, but then we get in experts in specific areas to come and spend the week with us. And every morning is lectures and discussion, and every afternoon is hands-on, working in the lab, learning how to do things like draw CSF or whatever. And then evening after dinner, we have a wine and cheese and talk about career opportunities and things like that. So it's,
Sure. But I fully enjoyed it and I learned a ton thanks to that workshop for sure.
Yeah.
So tell me the plans of what are you going to do this next 5 years with grant funding?
So the first 5 years was making a lot of these risk variants, and we've got over 30 different risk variants right now. And about 10 or 15 of those we have the data in hand, and the rest we're waiting. And this winter we'll be down, taking those mice down and doing the assays. So by next spring, next summer, we're gonna have a lot of data on the mouse models we have already, and we're gonna start making combinations of different alleles.
Okay.
And in top of the A beta and tau humanization, we'll put some of these, does PLCG2 work well with MTHFR, whatever the combination. And Greg Carter is doing bioinformatics to understand which of the disease modules, as shown by the AMP-AD portal, will be able to be combined to get more of a A-like phenotype. So we're making combinations, and these are going to get fully phenotyped up to 24 months of age. The other thing that's really new is being access to the humanized tau models. As I said, we have wild type, but we also have some risk variants in those.
Okay.
So those are new. No one in the world's characterized those yet, but we know that we can drive amyloid. We need to drive tau as well. So this is the essential step to that, and we think that'll get us a lot closer to it. So a big focus, one of the main aims of this is wild type tau, 2 different tau variants that have already been created, and we're going to be CRISPRing some new tau variants.
Okay.
2 or more, I should say. We're discussing with different people in the community who have expertise in this area. So we may be adding more tau risk variants of that as time goes on. And then again, we're combining these with APOE4, humanized A beta, and other
Keep adding
risk variants.
mutations on top of what you have already.
Not always just adding more and more, but different combinations.
Right.
Based on the omics that Greg Carter is analyzing. So we're really excited about that. What age is the greatest risk factor. Clearly, genetics is important, but environment is too. So we're working with high-fat diet. Or in different projects, we're looking at risk of ozone.
I saw that. Can you tell me a little bit more about the risk of ozone? I am not familiar with that.
Well, just
how it affects Alzheimer's progression.
Epidemiologically, it's been shown that if you live in a city and you're exposed to higher ozone levels, you're more likely to get Alzheimer's. So that's a clue for us. And people, our collaborators at Indiana University, actually have cages where you can modify put in some of these gases, and the mice are getting sicker sooner.
Is that right?
Certainly with that. Yeah. There's other things like sleep disruptions. Gareth Howell's group is looking at some heavy metals toxicity, which again has been correlated to Alzheimer's disease in patients. So there's lots of different options, and we're coming at this from all angles, from the genetics, the aging, and then the environmental factors as well.
That's so interesting.
It's a lot of work, and there's a lot of work to be done, but it's really exciting.
Yeah, but it seems like it's going on fantastic.
Yeah. I guess the other thing I should mention is we've been largely working on the quote-unquote vanilla black 6J mouse model right now.
Yes.
We do have some experiments putting some of these Alzheimer's alleles on different complex genetic backgrounds, and it's fascinating. We see very different results depending on which genetic background we put them in. So that's telling us a lot. That's telling us that genetics can play a role in driving these phenotypes, and now we can dig down into genetics and try to figure out what exact genes, what exact pathways, even risk variants.
That reminds me of the paper that Nonos wrote with the APP-PSEN1. It was remarkable differences depending if it was Black 6, PWK, and forget. Yes.
Yeah.
Yes.
Yeah, she's done a lot of great work on that. So that is in wild-derived inbred mouse lines.
Are you doing
we're doing
PhD?
we're doing collaborative cross.
Collaborative
mouse
cross.
lines. So
Okay.
it's a combined mixed genotype from 8 different founders, but they're reproducible. They're now inbred lines. There's Jack says 50 or 60, I think, different inbred.
That's right.
We picked some that have variations, and the risk loci we know are relevant for Alzheimer's disease, APOE4, PLC2, those things. So we're
Okay.
and it's really interesting. We're putting the same allele on these different CC lines. So it's very different. Amyloid, we're seeing different if the amyloid goes to vasculature versus parenchyma, which is
Okay.
a big part of human
Yes.
disease.
Yes.
And mostly we're seeing changes in microglial responses, just as Kristen Noble showed
Yes.
in her
Yes.
wild-derived papers. So we know that a lot of the genetic risk for Alzheimer's disease is not expressed in genes that are expressed in neurons, but they're expressed in genes that are expressed in microglia.
Correct.
So there's something about microglia that we know is essential for late onset Alzheimer's disease, and this gives us a handle to study those factors.
That's amazing.
Yeah.
That's very good. Now,
Yeah.
final question. What talks or what posters, were really, if you had to highlight something, what would you say?
There's so much going on this week. I guess Greg Carter's poster about the from our group again about the primary screening, where we had these variants which are correlated in human populations, and there's no, we can identify there's some association. We don't know what it is and why it is. The fact that we now have those in mouse models, and we're seeing changes in the proteomics and transcriptomics that mimic some of the things that we see in patients, that's telling us, but it's the first time we can get a handle on what these risk variants that are expressed in microglia are actually doing. And that's going to give us a hook to
It points to the translatability of these models
Exactly.
where
Exactly,
you don't
yeah.
see that in other
Right.
familiar models, perhaps.
Exactly, yeah. So if we can get a drug that reverses those changes in gene expression in the mouse models,
Right.
presumably those drugs would be not efficacious in humans, but they could be, the base model for moving forward and creating new drugs, or at least identifying pathways where we know what's targeting them. Because for so many years now, decades, we've just been looking at APP processing and amyloid processing. But again, the genetic risk is the microglial genes. So we need to understand microglia and astrocytes.
Absolutely.
But largely
Sounds
microglia.
like it's a response of the microglia to the plaques and tangles more than anything else, more than the presence of plaques and tangles by themselves.
Yeah,
In a lot of
and the
cases.
response, the microglial response, one guess is that it's very beneficial early when you're 40 or 50 and it's clearing out amyloid. But maybe when you're 70 or 80, the microglia are now actually attacking the synapse of the neurons.
Right.
So they go from being protective to being detrimental. So
Right.
it's really critical that you need to look at the right genes and the right pathways, but also at the right time. The
Right.
timing is essential. Because we
Interesting.
know this is a very slow progressing disease. And we want to look at those early prodromal phases, not the late phases.
So you've done so much work in the past 5 years. What time points do you think are the reasonable time points to look at these load models that you guys are making?
Well, I mean, it's always better to go later, but
But I guess my question is, what is the soonest that would make sense in a lot of these models that you're making?
yeah, I mean, like with the high-fat diet study, some of these are significantly different at 12 months of age, which
Okay.
is much more practical than waiting 24 months.
Yes.
But they're more severe phenotypes at 24 months if you're
And
if you're able to be patient.
do you see obesity in them or diabetes and things like that? Yes.
Oh yeah.
Similar
Yeah.
to a diet-induced, I guess it's like a diet-induced model
Yeah,
with
definitely, yeah.
risk factors
Yeah.
for Alzheimer's.
Yeah.
Okay.
Exactly.
All right.
And we know that, I mean, there's lots of evidence that cardiovascular risk is important in Alzheimer's disease, metabolic risk is important in Alzheimer's disease. So we're just trying to add those comorbidities into mouse models so we can better understand.
Does any of this research made you change your lifestyle in any way?
You shouldn't ask me that this week.
Well, you're very fit. You seem very fit. So
Actually, now that you mentioned that, all of the group, I was going to the course conference the other day, and two of the guys were running by me, two of the PIs were going out jogging. So yes, we're definitely trying to stay fit
yeah,
and active
Yeah.
and try to eat healthy.
yes,
Not so good at that.
that's amazing.
Yeah.
That's great. Well, thank you so much for
Thank you.
coming by our JAX booth and talking to you. And I'm very excited for everything that's going on with your group, and looking forward to updates later.
Yeah, we're excited to share.
Okay.
Yeah.
Thank you.
Thanks.
So here we are with Gareth Howell from the Jackson Laboratory, and we are live at SfN. And I just wanted to ask you about your research. What are the highlights of this past year that you wanna share with everybody? And I don't know, I'm sure you have a ton of posters. And what would you like to share with us? What's
Great. Well, thank you for the opportunity to tell you what we've been doing. So in the last year, we've obviously been working hard with creating new mouse models for late onset Alzheimer's disease as part of Model AD. We're just wrapping up a nearly 3-year study on the mice that carry ApoE4, humanized A beta, and humanized TREM2 mutation. And interestingly, we fed them a high-fat diet, which is a major environmental risk factor. And we call these mice load 2.
okay?
And these mice have a gene by environment interaction when we look at their phenotypes. So by 18 months, they look more like late onset Alzheimer's disease at the molecular level.
Okay.
And so we're really excited that we're beginning to see the emergence of late onset phenotypes in mice that haven't had amyloid driven by kind of familial mutations.
Right, because the humanized, I was talking to Mike Sasner before, and so the humanized beta is just it doesn't have any specific familial mutation. It's just a wild type.
Yeah, literally, we took mouse APP and we made 3 CRISPR edits to make the A beta sequence align to the human sequence. So we call that humanized A beta sequence. And so, yeah, no mutations, but we're beginning to see elevated levels of insoluble A beta when they're fed this high fat, high sugar diet.
Do you see that in CSF, or where do you see it?
We see it in blood.
In blood?
So it aligns quite nicely with what you see in human patients. I mean, we're definitely not there yet. We've got a lot more work to do in terms of driving the pathologies that we want, such as amyloid and tau. And so the next model that we're beginning to create, which we're calling Load 3, is humanized ApoE4, humanized A beta, but humanized tau. Again, all wild type, no mutations. And so we're really excited over the next couple of years to see what are the pathologies that might be emerging in this triple homozygous Load 3 mice. Again, we're going to use an environmental push. So we'll use the same high-fat diet, and we'll age them all the way out to 24 months, both here at the Jackson Labs, but also at Indiana University. And, you know, watch the space to see what these looks like. But we're already starting to do experiments in these mice. So, for instance, given the high-fat diet data, we're very interested in what other environmental stressors might do to these phenotypes. So even
like ozone, for example?
Like ozone at Indiana, at JAX, we're doing toxic metals. So we're all exposed to toxic metals during our lifetime. These are increased risk for things like late onset Alzheimer's disease. So these are things like cadmium, arsenic and lead. So these are in water, in the air. And so we're able to do studies in mice where we put those in the water.
Okay.
And either for an acute study or a chronic study, to really recapitulate what you see in, say, urban areas when you're walking to work and exposed to car fumes, or drinking well water, which is something we do quite a lot on the island at the Jackson Labs. And so, yeah, this is also potentially those of socioeconomic disadvantaged are more often exposed to these types of toxic metalloids.
Right.
So this is a great use of models like the Load 3 model and Load 2, because they don't show the pathologies. So the question then becomes, if we add an additional environmental risk,
are you kicking it over the
do we
edge?
push it more towards that risk? Exactly.
Yeah, that's great. And I know that you have done some studies, but I don't know exactly which strains with exercise and lack of exercise. Can you tell us a little bit about that?
Yeah, so earlier this year, my PhD student published a number of studies where she showed that there's an interaction between your genetics and the environment, such as exercise. So she used the ApoE mice, so humanized ApoE3 and humanized ApoE4.
All right.
But she did something a little bit different because of the mice that we created here at JAX. We're able to breed the mice together and create the most common risk genotype, which is actually ApoE3/4.
Okay.
And so previous studies of mice have often used homozygous ApoE3 or homozygous ApoE4. But we were able to generate these mice that were all 3 genotypes, 3/3, 3/4, and 4/4. And cutting a long story short, if you exercise these mice for up to a year, you see differences depending on what their genetics were.
Okay.
So there are unique changes, particularly in their brain transcriptomes, in 3/4s versus 4/4s, all compared to the neutral allele of ApoE33. So that
Was it
that
a
was
running
exciting.
wheel? What was
Yep.
it?
So we give them a voluntary running wheel. So
Okay.
they run as often as they like. And then periodically we put them in wheels where we can actually measure how much they run at night. So
Okay.
we can then track how much they ran, and so we could standardize those changes that we might see in
Well,
the
I
brain.
was curious to see any of those genotypes have a different preference for how much they run.
No, actually, that was an interesting question. We looked at that, and they ran as they were
Okay.
as motivated to run as each other. So
Okay.
genotype wasn't a factor in terms of how much they exercised.
And so what was the consequence of running for a whole year or not running as far as the
Yes.
phenotypes that you saw?
Right. So we didn't necessarily track specific AD phenotypes because they're not really present in the ApoE mice
Right.
using the assays. But we looked at things like cholesterol. So there's a cholesterol-specific changes. There are changes in particular pathways in the brain that have been associated with Alzheimer's disease, like innate immunity, particularly the complement cascade. So there's a unique change in some of the complement genes in ApoE3/4 mice that have been exercised you don't see in those other genotypes.
Okay, great. What other highlights do you wanna
Well, so the other thing we're working hard on is a lot of information about microglia and how microglia may be impacting Alzheimer's disease. And so a lot of single cell studies have started showing that there are different transcriptional states of microglia. And so in the lab, we've been working hard to start to understand what are the function of these supposed transcriptional states. And so these are microglia like disease-associated microglia that are associated with plaques or interferon-responding microglia. So in the next couple of years, we're going to be starting to track using genetics. If we ablate these microglia in their specific state, how does that affect the health of the brain, such as what happens to dendrites and synapses, what happens to plaques, what happens to cerebrovascular changes. And what's fascinating for us is that if you look at those microglial states in different mouse strains, so we've got amyloid on a variety of different genetic contexts, so wild-derived mouse strains,
Right.
they're different. And so just
You're
using
talking about like the APP, PS1 in different genetic
right,
backgrounds?
so we've induced amyloid in the regular B6 mice, but also in mice that are much more genetically diverse, such as the wild-derived strains. And so
Yeah.
these are WSB, PWK, and CAS. And so the numbers of disease-associated microglia differ across the
Yes.
strains, as do
I was just showing
interferon-responding
somebody
microglia.
else that the honors paper,
Yep.
and I
So
was
this is
showing the images. It's pretty amazing.
exactly. So this is work done by Kristen Onus in
Yeah.
my lab and Stanley Yang, a former postdoc. And so it's super exciting to think we can use these different mouse strains to begin to understand how genetic context changes their response, which we assume will be relevant to the genetic diversity you see in the human population.
Any tips for people working with the PWK that are so jumpy? We call them popcorn, at least the PWK inbreds. I mean, because I hear that it's challenging
It is.
to work with them.
Yep,
So,
they are popcorn mice, so don't take the lid off. Otherwise, they may well jump out of the box. We have special adaptations on top of the box. So we don't have an open box. We have plastic specially manufactured, so you can just go in and grab them out.
really?
Yeah, so we have really well-trained, mouse room staff and our own research technicians. Particularly, shout out to Kelly Keyser, who works with Kristen Onus. She's worked really hard to develop the right way of handling these mice so they don't become
Because I
stressed.
hear they just jump everywhere.
They do. They
You know,
do.
they go
Yeah.
behind the refrigerator, whatever. I mean,
Right. But what Kelly does, she stays very calm. I've seen it done. I'm nowhere near I can't do it like Kelly can. And so she just very calmly puts her hand into this specially manufactured box. And the mice just quite calmly jump on her hand.
Is that right?
And then she can transfer them very carefully. Yeah.
Oh,
So
my goodness.
lots of the research that we do would not be possible if it wasn't for the way Kelly was handling it.
Oh, wow. That's amazing.
Yeah. So don't shy away from them because you can learn how to use these mice, but they are different.
Right.
Feel free to contact us if you're really interested to use some of
Okay.
these
That's
these
great.
strains. Yeah.
I did not know that you had devised a special cage for them.
Yeah, Kristen Onus worked hard with some of the JAX engineering groups to make some of these adaptations. Not simply for the handling of mice, the cages, but also when we do behavioral assays. So we do a lot of Y-maze. You have to put covers on the Y-maze so that they don't jump out. When you put them to sleep, you have special adaptations to nose cones and things. So yeah, this is all stuff that we've had to work hard to manufacture at JAX. But we
Yeah.
can now use them like we use any other strain.
Amazing, amazing. So any talk outside Alzheimer's that you went at SfN that you know was
actually
striking to you?
Some of the work that we've been doing is now looking at similar microglial responses and changes in disease in the eye. So I'm particularly interested to identify those research groups that are working both in the brain and the eye, because obviously the retina is a piece of the brain that's sticking out of the skull, so it's easy to access. So there was a poster I was at this morning by Jason Meyer at Indiana University, who's using human IPS lines to basically understand the response of reactive astrocytes. And he can do this in cells that are cultured carrying mutations in glaucoma, which is another disease I study, but also Alzheimer's disease and also ALS. He can show differing responses depending on whether they carry the mutation. He can induce these astrocytes to become reactive using a special array of cytokines. So it's really interesting to be able to start thinking about shared mechanisms across multiple diseases. And we're doing that a lot in my lab as well. We've got glaucoma and diabetic retinopathy, as well as Alzheimer's and vascular dementia.
Awesome. So what's next in these 5 years that I know you have a lot of grant renewals.
We do. So Model AD.
What are you most excited about?
I'm excited that Model AD got renewed last year, beginning of this year. So that means we've got 5 more years to age more of the mice, to have different combinations. So it's a super exciting consortium. Love working with all the people there, such as Adrian O'Black and Bruce Lamb and Paul Torito, Stacey Rizzo at Pitt. But the other thing we've got recently funded was to question whether the retina is a good biomarker for Alzheimer's disease. So there's a lot of data out there that suggests it is, some data to suggest it's not.
Right.
And so we're going to be using both some of the strains that we've created in the Howell lab, such as the wild-derived strains that I talked about,
but
also the Model AD strains, to find out what are the changes that are happening in the retina that may be reflected of changes in the brain.
So what do people claim to see? Plaques in the retina?
Some people claim to see plaques. I think we're as excited to determine whether you see the vascular changes that you might be seeing in Alzheimer's disease and related dementias, maybe particularly kind of vascular contributions to what's called vascular contribution to cognitive impairment in dementia, or VCID. So we have a number of strains that we think of
there's a lot of overlap.
More appropriate, right? Exactly. So my postdoc Elena Reagan published a paper earlier this year on a variant in the MTHFR gene, which is in folate metabolism, methionine metabolism. And you can see both brain changes to the vasculature, but also changes in the retinal vasculature. And this is a risk variant that has been associated with a whole variety of different diseases, including dementia, but also glaucoma, cardiovascular, cancer. So we now have a mouse model that we can start asking questions about how this relatively common variant, which maybe 20 to 40% of us carry at least one copy, how that increases risk for a variety of different diseases. So the eye as a biomarker is something that we're really focusing on.
That would be fantastic.
Yeah,
because the problem is that by the time they diagnose it, it's a little bit too late. Nowadays.
Yeah, so there's a lot of work in blood. So obviously blood biomarkers is something that's studied a lot. I think where we think is to really identify those earliest changes, you may be able to combine the data in the eye with the data in the blood.
Nice.
So we're aligning our blood-based biomarkers partly through Model AD in collaboration with Jeff.
And those biomarkers, I saw the list. I mean, they're the same biomarkers that are used in clinical trials.
Almost
what I love about your consortium is that it's really aiming at translational research.
Very much so. So that when we're ready to take a mouse to do a preclinical study, we're already using the same things that would be used in a clinical trial.
That's remarkable, including brain MRI and all of that.
Exactly. Yep.
That's amazing.
Yeah. So it's exciting times ahead.
Yeah, that's great. Well, thank you so much for stopping by, and
no
thank
problem.
you for sharing all the good stuff that is going on in your lab. And good luck with the next 5 years.
Great. Thank you.
Thank you.